Thank you. Yeah, I really appreciate the opportunity to be here and chat to you guys about the work that we're starting. Am I standing or meeting?
You're fine.
Okay. About the work that we're starting and hear RJ and Bill's information about other projects that are going on. So I'm Laura Beecraft. I'm in the lab of Dr. Michael Wetz at the Harte Research Institute for Gulf of Mexico Studies. And our work focuses on water quality in the different bays and estuaries along our coast here. And I'm going to be talking about assessing the risks to ecosystem health from nutrient pollution in Lavaca Bay. So nutrient pollution, when we're talking about it, we usually mean nitrogen and phosphorus. I'm sure most of you have heard those elements. They're fertilizer, they're essential for all plants, including the microscopic plants in the water, the phytoplankton. So we need them coming from the land into our freshwaters, into our coasts, but as is often the case, too much of a good thing becomes a problem, and then we refer to it as nutrient pollution, and it can lead to deterioration of ecosystem health in a variety of ways. One of those is supporting excessive phytoplankton growth, which we can then call a bloom. Not all blooms are bad, but they can become harmful algal blooms when they have adverse effects on the ecosystem or on humans, through a variety of mechanisms like toxin production, decreasing dissolved oxygen, irritating fish and bivalves through physical irritation, mucus production, surfactants, other biochemical compounds that they can produce. They decrease light penetration in the water column, shading out seagrasses and other thick vegetation. And then as they're breaking down, the bacteria that start to degrade them are pulling oxygen out of the water and decreasing it, creating hypoxic or anoxic conditions that can adversely affect other organisms. And incoming nutrient pollution is often associated with watershed land use, things like urbanization, agriculture without mitigating best management practices, water treatment or septic systems that maybe aren't up to snuff or are under a lot of pressure, and all these sources, but we've got natural sources too, like wetlands. So I'm going to start off with a bit of observations from a recently completed and an ongoing study in the Matagorda Bay ecosystem. Both of them have sampling sites within Lavaca Bay. And the first one is a water quality trend analysis of the Matagorda Bay ecosystem funded by Texas Comptroller, and it used data from the Texas Commission on Environmental Quality's Surface Water Quality Monitoring Program. And it involved a trend analysis over a 10+ year period, as well as comparing different water quality parameters to TCEQ screening thresholds. So they were looking at things like salinity, pH, temperature, dissolved oxygen, nitrogen and phosphorus, and chlorophyll A concentration. And some of you may have heard chlorophyll A. If you haven't, it's the pigment that's in all photosynthesizing organisms, and we use it as an effective way to estimate the phytoplankton or algal biomass in the water. So just a few results from this trend analysis. We've got some heat maps here. The color gradient in the bay is showing the relative concentration of the respective parameters. So the cool colors, the dark blue, mean a low concentration, and then as the colors warm, it means an increasing concentration of that parameter. In this case, we have nitrogen, total Kaldol nitrogen on the left, and total phosphorus on the right, two nutrient parameters measured as part of the TCEQ monitoring program. So we can see that both of those nutrients are a little bit higher in Lavaca Bay compared to the rest of the Matagorda system. And then the other aspect of these maps are the specific sites themselves, and the symbols are related to the results of the trend analysis, that time series they looked at. And the black squares mean there was no statistically significant change in that parameter over that time period. The red circles mean an increase in that parameter over time, and blue circles a decrease in that parameter over time. So they found evidence of increasing nutrients in parts of Lavaca Bay and western Matagorda Bay for nitrogen and phosphorus. And I'll mention that the TCEQ data is generally quarterly sampled, and these heat maps are just based on the sites that are marked, and the color in between is kind of interpolated by an algorithm. So it's just based on the different site points that we see here. And next we'll look at the chlorophyll and dissolved oxygen that I mentioned. We can think of these as nutrient indicators because they tell us something about how the system is responding to the nutrients that are coming in. And on the left, we've got chlorophyll where there was low to moderate levels throughout the system, but you'll notice there's a couple spots with red points, so there was evidence of localized long-term increases in chlorophyll concentration in Lavaca and Western Matagorda. And then on the right-hand side, we've got some blue dots indicating long-term decreases in dissolved oxygen in a few locations. The yellow on the dissolved oxygen side is nothing to worry about. We want higher dissolved oxygen.
So, Laura, both of those are beneficial in increasing amounts, right?
Chlorophyll's a little give and take. We want some in there, but we don't necessarily want too much in there.
Okay.
And then sometimes it depends which species of phytoplankton are part of that chlorophyll, which we'll touch on in a little bit. Dissolved oxygen, we do want higher. We don't want it decreasing. So in the nutrients and the chlorophyll, the red dots are not ideal, and in the dissolved oxygen, the blue dots are not ideal. And next, I'm going to show just some preliminary results from a study that's still wrapping up. This was part of a water quality and harmful algal bloom monitoring program funded by the Lavaca Bay Mitigation Trust, and it involved weekly sampling at 6 sites, and 2 in San Antonio Bay, 4 in Matagorda Bay, one of them just off the Bayfront Peninsula Park here. So that's the data I'm showing you, just from that site that we referred to as our Port Lavaca site. The top graph is showing ammonium concentration. That's a form of nitrogen that is readily available and used by phytoplankton. And we did see some pretty dramatic peaks in ammonium concentration from the samples we've analyzed so far for this study. And then on the bottom, we have chlorophyll concentration. So that's our phytoplankton biomass, really variable, but again, we had some pretty high peaks, and over that 2-year period, 28% of the samples we collected exceeded the TCEQ threshold for impairment.
Wow.
During this study as well, something else we did was look at those samples under the microscope the same day they were collected to see who's there. We were specifically, or in particular, looking out for any harmful algal bloom events or the presence of species that can form harmful algal blooms. So kind of a qualitative screening of the phytoplankton composition. What we found here with our Port Lavaca site, diatoms on the left-hand side are what we see most commonly in the most of, a whole bunch of different kinds. I'm just showing 4 different species here. Diatoms are the ones that have a little silica frustule. They build a little glass box around themselves, and we usually think of them as good phytoplankton. They're ones that are preferred and beneficial for consumers, and they produce higher amounts of fatty acids and essential fatty acids that are important not only for their immediate consumers, but the whole food web, because essential fatty acids can be produced by the higher consumers. But we did see a few species of concern. They weren't at harmful concentrations, but they are occasionally there. And 2 of those were the dinoflagellate Apachila sanguinae, and we couldn't fully identify a rhapidophyte, but it's part of a group that has a lot of toxin producers. So nothing to worry about yet, but we did see these there. So kind of pulling stuff together from those 2 studies and from that water quality trend analysis, we didn't observe evidence of any issues right now, no frequent HABs or hypoxia or fish kills, but there was evidence that certain trends were creeping upwards that we don't want to see. And in particular, they were more common in Lavaca and Western Matagorda, that decreasing dissolved oxygen, increasing nitrogen and phosphorus, and increasing chlorophyll. So some recommendations that came from that report on the TCEQ data were targeted data collections to validate the trends that we're seeing, because again, that TCEQ data is quarterly and at a smaller number of sites. And we also want to understand the drivers behind those trends we're seeing. And then potential implementation of best management practices or watershed protection plans to help address those nutrient sources if we know where they're coming from. So that leads us into the reason for the study we're starting now, assessing the risk to ecosystem health in Lavaca from increasing nitrogen and phosphorus. 2 main goals that we have for this are to determine nutrient water quality conditions within the bay, with the focus on assessing its sensitivity of the phytoplankton communities to increasing nutrient concentrations. For this first goal, what we're really looking to do is create a more temporally and spatially comprehensive picture of the water quality in the bay. And this will be done with monthly sampling at 8 fixed locations chosen to kind of capture a variety of inflow sources into Lavaca. And we're hoping to identify any possible sources of nutrients, different types of nutrients into the system, and drivers of phytoplankton growth. And this is a part of our project that if there's community members that are interested in getting involved and have availability to take us out on their boat and drive that boat for our monthly sampling, that would be fantastic. So if you or you know someone that might be interested, please reach out or get my contact information from Janet. That would be fabulous. And then we'd also love to get, you know, the benefit of your knowledge if you've been around Lavaca all your life or for a longer period of time, and that is great too.
While you're there, just
Yes.
for the heck of it,
Yeah.
there is a lot of farming activity all along the shoreline, particularly up here. You don't have any sampling areas there, and there's a lot of runoff right there.
Right, direction.
Yep.
Are there any large streams that would help kind of concentrate?
Yes, there is.
There are some streams.
There's
that
one
you can
particular
target?
real bad erosion project going on,
Okay.
and there's no telling what all's coming out of there from the fields.
Okay, thank you.
It's right about where that little point sticks out
All right.
in the dark spot.
Okay, I'm gonna pester you about that later. Thank
Okay.
you. Good. I was wondering, because we have a big gap there, but just, again, I'm also, I don't know if you can tell if I have an accent. I'm originally from Canada. I've been in Texas less than 2 years. I came down here to join Dr. Wetz's lab. So my local geography leaves something to be desired. So,
If you want an idea on pollution sources that are coming into the bay, put an information request into the Department of State Health Services Seafood and Aquatic Life Group and ask for the Lavaca Bay sanitary survey of the shellfish harvesting areas, because everything's in there, from industrial effluent to wastewater plants to crops, major drainage ditches like Maxwell Ditch. I mean, it's all covered in the comprehensive sanitary survey, and it may save you a lot of time.
Great idea.
His name is R.J. Shelley. You don't have to remember all the others. R.J. Shelley.
RJ's already been putting up with some of my emails too, so I've got that. Thank you. You guys are gonna make me want twice as many sampling sites, and that's gonna be a logistical problem. I'm gonna have to pick the best ones, or we're gonna have to pick the best ones. And for our second part of this project, second main goal, assessing the sensitivity of the phytoplankton to increasing nutrients, we'll be doing this with experimental bioassays. Bioassays, simply put, are an experiment using some biological organism to assess a response to sun condition. And we'll be taking bay water close to one of our monthly sampling sites, bringing it to a near shore location, and setting it up for a 72-hour experiment that we can access on foot from shore, so we can keep an eye on it and do regular sampling from it. It'll essentially be a whole bunch of clear containers of water, and then they'll go in a little corral so they don't get away. They'll be exposed to natural temperature and light conditions, but we'll add different forms of nitrogen and phosphorus to try and identify which nutrient and form elicits the largest response in the phytoplankton. That's year 1. And then in year 2, we're going to pick the one that had the biggest response and kind of be creating a concentration gradient and try and see if there's a threshold or a point at which the phytoplankton community, at least, is kind of tipping into an ecological state that we don't want to see. Trying to identify if there's a break point that we need to know about, hopefully well before we get there. So that's what we're aiming to do with this project. I'd just like to thank Lavaca Bay Mitigation Trust for funding the project that's ongoing and the one we're about to start, and Texas Comptroller for the analysis of the TCQ data set. Thank you for your time.